All right, so for Lab Practical 2, we need to go over the last of our three classes of tissues. Practical 1, we already did the connective tissues, so our last three are going to be epithelial, muscle, and nervous tissues. So let's start off with the epithelial tissue. All right. First off, it's really important that you guys remember that when we're talking about epithelial tissue, it's always tissue that's at the boundary of the body. So think it's at the very surface of the body, like in the skin, or any place that's lining and opening, like the inside of the mouth or the trachea. So when we're looking for epithelial tissue, we're always going to look on the border or the boundary of the tissue. That's important because these are slides made from real tissues, slices from organs. And organs, something like the stomach or the esophagus, are generally made of multiple tissue types. So when you just look at one of these slides, you're going to probably see a couple different tissue types there. And so if we're going to find the epithelia, we want to look at those boundaries. Again, epithelia, any place that we're on contact with the outside world. So the outside of the skin, the inside of passageways or tubes. Our first epithelial tissue is a simple squamous epithelia. All right, here we're going to actually look at these tissues sort of top down. So you're looking at the top of these cells. One of the really sort of easy ways to immediately distinguish simple squamous epithelia is that they kind of look like a whole bunch of fried eggs put together. Like if you kind of imagine cracking a bunch of eggs in the pan and they all kind of spread out and like there's a yolk. They all have sort of this irregular border and all the cells just reach out until they touch the cells nearby. Simple, hopefully we remember from lecture, means that there's just one layer of cells, so this tissue is one layer thick. Squamous means flat, so the cells are flat. So this is a single layer of flat cells. When we're looking at these cells, again, irregular border, and we have sort of this fried egg look with a dark nucleus and then sort of all this lighter space inside. For this class, you need to be able to look at this tissue and immediately know it's a simple squamous epithelia. We also have a couple structures we need to know. First off, the cell membrane. And so here we're pointing at the cell membrane. The cell membrane is just the boundary to each and every cell. So you just kind of look and you can see that there's this line that surrounds each cell, sort of like, lining the edge of a fried egg. That's all cell membrane. The nucleus is going to be the dark spot in the middle of each cell. So if this is a fried egg, sort of the yolk looking thing. The way these tissues are stained, pretty much every cell we're going to see is actually going to have the nucleus stained as a dark spot, usually in the middle. So the darkest spot you see inside any cell in this class is going to be a nucleus. And then finally, cytoplasm. And we just define cytoplasm as everything in the cell that's between the cell membrane and the nucleus. So all of this more lightly colored, sort of the egg white, if it's a fried egg, all of that cytoplasm. So you can see we're pointing to it right here. Okay, so pretty easy to distinguish these structures and this tissue. Now we're going to take a look at a stratified squamous epithelia. Still flat cells, but now that it's stratified, that means that these cells are stacked up. There's layers of them. Right? Here we're actually looking on the side. So this would be sort of the outside space. And then here actually we have some connective tissue. And so you can sort of see there's all these layers of cells stacked up one on top of each other. So it's quite a few layers thick. A lot of students get confused because we say this is a squamous epithelia, and the cells are actually easiest to distinguish down here. And if you look down here at the very bottommost layers, these cells aren't that flat. They actually look kind of round or maybe even kind of cuboidal. All right? The way we're going to distinguish that this is a squamous tissue, though, is we need to actually look at the outermost layer of cells. Okay, so the cells that are actually touching this open space, these cells look flat. They look very squished and smashed down. Since these cells are flat, we're going to classify this entire tissue as being a squamous epithelia. Okay, I'll switch back to this when we look at transitionals so you can sort of see the difference. Just like with our simple squamous epithelia, many of the structures we need to know are the same. Cell membrane is going to be the boundary around each cell. Right? And so yeah, it's kind of hard to see some of these membranes, but you can make them out on a few different cells. Our nucleus is always going to be that dark spot that you can see in the middle. Cytoplasm, everything else. But now we also have a new structure called the basal membrane, oftentimes also called the basement membrane or the basal lamina. Remember, we've stacked up all of these cells to be multiple cells thick, but there is a very first layer of cells. And laying all underneath that very first layer of cells, I've sort of drawn it out for you right here, there's going to be a membrane that we sort of attach all of these cells to. That's the basement membrane. So it's this line, sort of this border between all of this epithelia and then this connective tissue, which is actually underneath. Okay. Now we're going to look at transitional epithelia because it looks very similar to stratified squamous. All right. Transitional epithelia is going to have lots of cells stacked up, so it is a stratified tissue, right? And you can sort of see down here, especially at the very bottom of the epithelia, the cells look very similar to the way they looked here in stratified squamous. The major difference between these two is going to be looking at these outermost cells. So again, we're looking at the cells closest to this outside space. These cells are not very flat. You can see pretty clearly these cells are sort of round-ish, quite irregularly shaped, okay? That means that this is not a squamous tissue. This is going to be a transitional epithelium, okay? So it's going to be the easiest way to tell transitional and stratified squamous epithelia apart. Just look up at this outermost layer. All right, again, these cells are pretty irregularly shaped, kind of fat, like a cuboidal cell, but have all of these very odd boundaries, which makes them a little bit more like a squamous cell. So they're somewhere in between. Remember, this tissue is generally used in organs that need to stretch quite a bit, particularly the urinary bladder. Structures we need to see, just like with our stratified squamous epithelia, cell membrane is going to be all of this border around each individual cell. And so this picture is a little bit nicer. You can see more of these borders here. Dark spots in the middle are all nuclei. Our cytoplasm, all of this clearer spot are between the nucleus and the cell membrane. And then again, the basement membrane. So if we go, here's the outside space. This would be the outermost layer of cells. We go all the way down to the innermost layer, all right, where this sort of the last layer of cells that all look the same. And then we're going to see that there's this line that separates the tissue, the epithelia, from the connective tissue below, and that is going to be our basement membrane. Now our simple cuboidal epithelia, again, simple means a single layer. Cuboidal now, these cells are going to be a little bit more cube-shaped, so sort of square-shaped. All right, and in general, when we see this tissue, they tend to be found in tubes or in ducts. So it's really common to see them arranged in rings. And here you can see there's all these different rings of cells. All right, it is epithelial tissue, so it's going to be tissue that's bordering space. And so here the space is actually all of this open space inside of these tubes. And then the structures we need to know are generally the same as what we've seen before. The cell membrane, all of this border around each individual cell, and these cell membranes are stained really nicely. Dark spots in the middle are all nuclei. All of the space in between are nuclei and our cell membrane. So here's some, that's cytoplasm. And then finally the bottommost layer, right? That means this is epithelia, so the innermost side is going to be touching the space. So the opposite side, this is the bottom layer. This is where we're going to find that basement membrane. And it's sort of drawn out for you a little bit here in blue. Simple columnar epithelia now. Simple still means a single layer, but now those cells are columnar, which means shaped like a column, so think tall and skinny. And the structures we need to see, again, very similar initially. So we've got cell membrane. So here we're looking at all these cells, and here is this epithelial tissue right here. Okay, so you can see all of this open space. Easiest way to find epithelial tissue is find the open space, and then the epithelial tissue are the ones that are actually touching it. So this is all our epithelial cells. Our cell membrane, as always, is the line surrounding each cell. And you can see these cells are kind of tall and skinny. They're a little odd shaped. Here's a really sort of nice example of one of these tall, skinny cells right here. All right, so there's its cell membrane. Dark spot in the middle, still nuclei. Cytoplasm, all of the space in between nucleus and cell membrane, so this lighter space. Now we have two new structures. There's microvilli, and then there's the brush border. Right. Microvilli, maybe you guys remember, they're small projections of the cytoplasm. So let's make a pen. I don't know what color my pen is. Oh, good, it's white. All right. So if we look at these cells, the cell is kind of tall and skinny. There's the cell membrane. I'll draw a nucleus right there, and then there's all the cytoplasm. On the apical surface, so the surface closest to the space, what you'll actually find is the cell membrane makes these little almost kind of hair-like projections. All right, so this is from chapter 3, so hopefully we remember this, and they're called microvilli. These microvilli are really useful for increasing absorption inside these tissues. All right, so you get sort of this fuzzy surface on these cells. The microvilli are each of these little projections on that apical surface. Because our microscopes are only, so powerful, the microvilli are all here. All right, and if you could zoom in really closely, you would see that there are lots and lots of these little sort of hairs brushing off of each cell. However, on these microscopes, we can't zoom in that well, and so what we actually see is this sort of fuzzy border. And so you can, I think, tell that there's a little bit of loss of focus here. Something's going on here, but we can't actually see it. That's what we're going to call our brush border. And it's called a brush border because if you sort of look at it, it's made up of all these little hairs and it kind of looks like a brush. Okay, so we can't actually see those microvilli, but we can see that brush border. But you should know what both of those structures are. Inside this epithelium, we also will commonly find another type of cell called a goblet cell. These goblet cells are pretty large, really quite round, and these are generally mucus-producing cells. These simple columnar epithelia we tend to find in passageways where lots of things move through, like your intestines. And so goblet cells will be there to produce mucus and lubricate this entire surface as well. Now our pseudostratified columnar epithelia. Pseudo means false. Stratified, as we know before, means arranged in layers. But so these are false layers. What that means is if you look at this epithelia, the cells look like they might be in multiple layers, but they're actually just one layer of tall, thin cells. Right? And so what I mean by that is you kind of look around here, and you can see this dark spot that's a nucleus and this dark spot that's a nucleus. And there's sort of nuclei that are high and there are nuclei that are low. And that can commonly make it look like there's actually 2, maybe even 3 layers of cells. But what we'll actually see is if you kind of look at these cells, they tend to have sort of this irregular shape where maybe one cell's fatter on the top and so its nuclei is high, and then another cell is sort of fatter at the bottom, so its nucleus is low. And it makes it look from sort of far away that there's a row of cells up top here and then a row of cells on the bottom. But that's not actually true. The way I'm drawing it, you can sort of see that all of the cells contact the very, very top and the very, very bottom. They're just sort of staggered in that appearance. So we say that it looks stratified, but it's not actually stratified. It's pseudo-stratified. All right, and columnar, again, these cells are tall and skinny. Just like before, we need to know the cell membrane, which is this border around each and every cell. And so you can see some of these cell membranes right here. Dark spot in the middle, always the nucleus. Cytoplasm, always the space between the nucleus and the cell membrane. Right. Goblet cells will be in this tissue as well. Here's a goblet cell right here for producing mucus. And now, though, we have these projections on the top. All right, they look in sort of up close a lot like microvilli. Except these projections are called cilia, and they're much bigger. So when we looked at microvilli before, you couldn't actually see each individual microvilli. You only saw sort of this fuzziness. When we look at these projections here, you can actually see each individual little hair. That's because these are much, much larger. These are cilia, okay? So cilia we can actually distinguish. Generally, the role of cilia is they move and they create a current and can sort of beat fluids or mucus along this surface, right? So they're going to be a lot larger. Also, as we've said before, basement membrane, this membrane on the very bottom of our layer of cells. All right, that's our epithelial tissue. So nothing too complicated. Now we can talk about our muscle tissues. First off is our skeletal muscle tissue. So these are the muscles that are actually attached to your bones that let you move. The structures we need to know. Well, first off, you look at this tissue and you can see it's got all these, these cells are sort of arranged in nice little parallel lines. It's got this really sort of characteristic striped pattern to the tissue. Each of these long, thick bands is what we're going to call a fiber. A fiber is just a cell. And so hopefully we remember from lecture that skeletal muscle cells are very, very long. So long, in fact, that they tend to resemble fibers. So here's one fiber, this is another fiber, you can see it right here. Here's another one there. So all of these long, thick lines are fibers. Right? These fibers are also striated. Right? If you can see that this fiber is running sort of up and down in this direction, that's the way the cell is running, then you can see all of these little stripes that run parallel. Right? These are all striations, and you can see I've sort of dotted out a couple for you here. Striations, those are because of the way the sarcomeres are organized, and that's what actually allows for contraction, and skeletal muscles will be striated all the way through, and so you can see these stripes all running perpendicular to the fiber. Skeletal muscle tissue also has nuclei. These cells tend to be multinucleated, so there will be many nuclei per cell, but in these cells, rather than being in the very, very middle of the cell, the nuclei are actually going to be off on the side of the fiber. So these dark spots that are right at the edge of the cell membrane, these are all nuclei right here. Okay? And then finally, connective tissue. Oftentimes, between fibers, you'll find some extra tissue sort of packed in between, holding them together. Usually it's adipose tissue, some fat sort of packed into that muscle. And so here's some connective tissue, this just sort of open-looking space. Here's some connective tissue here. This is probably a little bit better. You can actually sort of see some of these cells. Cardiac muscle tissue, on the other hand, looks really quite similar, but there's one major difference. These cells are not nearly as long, right? So the individual cells or fibers are much shorter, and you can sort of find the borders of them. So sort of here's one border, and then this cell runs around here. So there we have sort of one cardiac muscle cell, and I just draw it. All right. These cardiac muscle cells are still striated, so you can see sort of faintly there are these light purple lines that are running perpendicular to the fiber. All right, we still have a nucleus, a dark center. Now those nuclei do tend to be a little bit more centered inside the cell. Connective tissue will still be just this tissue that's in between these purple cells, sort of holding them together, filling in spaces. Now we also have something called an intercalated disc. The intercalated discs are these dark, sort of jagged lines that connect two different cardiocytes together, or two different heart muscle cells together. So we can see there's lots of these intercalated discs. They're basically structures that stitch these cells together very strongly. Our last type of muscle cell is a smooth muscle cell. These ones are pretty short and also generally really quite spindle-shaped cells. And what I mean by spindle-shaped, spindle means that it's sort of pointed at the ends and kind of fat in the middle. So the cells do generally look something like that. All right, and they all have a nucleus in the middle of them, usually. All right, so cell membrane, again, all of this space around the cell, and so you can sort of see this cell membrane around these cells. Nucleus, all these dark spots in the middle, and then cytoplasm, everything between nucleus and our cell membrane. So let's see, sort of, here's a pretty decent spindly shaped cell. It kind of goes like this. It's fairly lightly stained though. All right, and then our last type of tissue that we need to know for this second practical is our nervous tissue. Nervous tissue's got a pretty distinct look to it. All right, we have these really, really big blue cells that have sort of like spider webby type projections, and then lots of much, much smaller blue cells in the background. All right, there's actually two different types of cells that we need to know on this tissue. The really, really big ones, these are our neurons. All right, these small little guys in the background, these are our neuroglia. And hopefully we also remember from lecture the neurons are the ones that are actually sending and receiving signals and impulses. The neuroglia are support cells, so they are sort of supporting and helping these large neurons to function. All right. Within our neuron, we have a couple pieces that we need to know. The cell body is this large inner portion. So here's the cell body as well. Right? Sort of this just center portion, generally where you're going to find that dark spot, the nucleus. Now you have all of these little spindly things coming off the cell. We call these broadly cell processes. In lecture, you did learn that there were two different types of cell processes, axons and dendrites, right? And functionally, those are very different. But with this microscopy and this level of staining, it's really difficult to tell the difference, and in fact, you actually can't on this level. So if you were to just call one of these processes an axon, well, that might not be totally right. It could be a dendrite. You don't actually know. So to call it a cell process is generally accepted as being correct and, not being overly exclusive. And then finally, those neuroglial cells are the ones down here that are just all sort of around. You can see all these little dark blue nuclei filling in the space around these neurons. Right? And so those are the tissues we need to know for lab practical 2. Again, you all need to be prepared for the practical to identify any of these tissues on site. So if we showed you these tissues, you should know what they all are. Be very specific. So if the answer is smooth muscle tissue, you have to say smooth. You can't just say muscle tissue. Right? Not only do you have to identify the tissue type by site, but you also need to identify all of the different structures that we've talked about. So if a question was, name this structure right here, then the answer would be cell process. So you just need to prepare and go over these images, draw them, study them, familiarize yourself with them, and you should be in good shape for that portion of the lab practical. All right, good luck.